Thanks for joining us again for another biology lecture. Today we're talking about mitosis, which is a type of cell division where one cell divides into two genetically identical cells. Replication within our bodies is necessary as old or damaged cells have to be replaced. Replication is also required for the formation of organisms, as cells are the foundation of every living organisms. Mitosis is part of the cell cycle, with the paramount goal to generate two identical cells from one mother cell. For organisms which replicate in an asexual way, it's also the way to generate genetically identical offspring. In our body there is a process called meiosis, which we'll discuss in another video. Mitosis needs to be performed flawlessly as an uneven distribution of chromosomes. So chromosomes are the highly condensed form of DNA, wrapped around proteins basically. This can lead to cell death or cause diseases like cancer. Mitosis is a part of the cell cycle. And as you can see here, it's only a small part, but it's a very essential one. It can be divided into four main phases, which are Prophase, Metaphase, Anaphase and Telophase. Prophase can be divided, and some teachers may do that, into Prophase and Prometaphase, and we're going to discuss that as well. Of course, before we can start mitosis, it's necessary to duplicate the genetic material. So these are steps that are done before in the cell cycle. We're going to talk about the cell cycle in another video as well. Now let's talk about the phases in more detail. So in prophase, the chromosomes, so the DNA is starting to condense, and the mitotic spindle, which is then responsible for pulling the chromosomes apart, forms. Nucleoli, which are responsible for producing ribosomes, so the protein factories within our cells, they disappear, and this is a sign that mitosis starts. And this is what we have called prophase, and now prometaphase is that the condensation of the chromosomes advances, so they're even more condensed, and the nuclear envelope starts to break down, while the mitotic spindle connects to the kinetochores, so these are part of the chromosomes where the mitotic spindle connects to the chromosomes. Then we're on to metaphase. There the chromosomes have to align in one line, which is called metaphase plate or equatorial plane. And this is the first step for pulling the chromosomes apart, because it is very essential that they are pulled apart equally. And there's also a checkpoint, the metaphase checkpoint, and only if all chromosomes are aligned within the metaphase plate, the whole process continues or can continue. In anaphase, the cohesines, which are responsible for holding the two sister chromatids together, they are cleaved, and the chromosomes are now pulled apart, and they're pulled to opposite sides of the cell. This process is called out by motor proteins, which are called kinesin and dynein, and by this process the cell also gets longer. In telophase, the cell expands even more because it has to prepare for the separation of the two newly formed daughter cells. As we discussed before, the nuclear envelope broke down and the nuclei disappeared, and now they have to come back again, they have to be formed again, and the chromosomes start to decondense, and the DNA is now back into its linear form. Cytokinesis, which is the actual process of separating cells, can start during anaphase or telophase, but this depends on the organism. So in animal cells, like in our cells, there's a band of filaments which disentangles the two cells, and this starts during anaphase or telophase. But for example, organisms like fungi, mitosis and cytokinesis are two separate processes as they've got multiple nuclei. So, and now we've got 2 new cells. They're capable of surviving, they can start to divide again, they can go back into T0 phase and just like continue to live. Thanks for watching, we hope you liked this video and see you in the next one.